what is the key differences between XRD and grazing incident wide angle X-ray scattering. Both techniques are used for structural analysis. XRD is basically used for bulk material. This is the key difference. While this technique, which is specified techniques, used for thin film, So why we use this for thin film? Because in this case, we use very shallow incident X-ray angle, that is around 0.2 degree, which is very low angle. So with this incident angle, the X-ray cannot penetrate to the bulk material. It will only study the surface. While in the case of conventional XRD, which is used for bulk material, we can change the incident angle. So because of that, the incident X-ray can go inside the material. I will also explain how to write about these two techniques, and these techniques must support each other. Generally, we use these two techniques to analyze, to know about different parameters. First of all, we want to see intensity enhancement or reduction. This is one clue, one indication from results. So in conventional XRD, we have peaks here, and this peak is basically one-dimensional peak here. So when we have increase in peak or decrease in peaks, so it gives us information. In the case of grazing incident angle, we basically get ring here, and that ring is basically two-dimensional. So when for example, the second important thing is when we have new peaks or ring here, ring appear. This is very important. So we can see that new phase is formed. In some cases, we have disappearing new peaks or ring. So peaks in the form of conventional XRD, while ring in the form of grazing X-ray scattering, right? So when some new peaks are appearing, so we say some phase is forming. When some peaks are basically, or when some peaks, not new peaks, when some peaks are ring are disappearing, so this means that basically the unwanted residue or those phases are basically disappearing. Another important thing is the increase or decrease in full width half maxima in the case of one dimensional. Let's get started from the conventional XRD. This is basically the XRD graph here. So we have two samples. This is one sample and this is another samples. So when the sample is fabricated, so this is the initial XRD. So when they took the XRD, after that they kept the sample for 2000 hours in nitrogen box to see what is internally changing, whether the structure remains the same, stable, or whether the structure is changing. So we can see here that initially there is unwanted peak. This is basically the lead diiodide peak here. And I have shown here, this is basically the XRD peak for this lead diiodide. And this is the main peak here. It is about 12 2 theta, somehow 12. So we can see that this is the same peak here. So this is the unwanted peak here. So what happened when they kept the sample for 2000 R? So that unwanted peak increases here. This means that the material is degrading with the time here. This means that the amount or the phases for this lead diiodide is increasing. How we know? We know from this peak intensity here. You see, this is unwanted peak here. Now what happened here, when the surface is modified with another material, so we can see that this lead diiodide is by default there. So that peak is basically disappear, the lead diiodide, because of this additional modifier here. When they kept the sample for 2000 hours, again that peak is not appearing here. You see here, there is no changes, there is no additional or new peaks appearing. So this means that this material is basically signify, enhance the crystallinity and remove those unwanted species from the sample. And this kind of information we can know from XRD analysis. Now let's compare it with the grazing incident wide angle X-ray scattering. So here we have the two sample. This is basically the first sample here, and this is the second sample here. And this is the main peak here. This is the 100 peak here. So we can see here that that peak is basically visible here in both cases, yes, but in the first case, just like this lead diiodide peak here, so that small peak is also appearing here. You see very clearly, while that peak is disappearing here from this sample, and we can see that there is no dead peak here. So this is how these two techniques support each other. So these graphs I took from, this is the paper I took the graph from, and it is published in Nature Communication. This is the title of the paper by Yihu Wing et al. In 2024. This paper is basically published under a common license 4.0, and this is the link to the license.